We inspected them and got them here. We have meticulously selected the workmen and trained them to international standards. Every single pill which is manufactured and packed, it is our commitment, our responsibility as a team. We should give the customer a quality product. I want to be in this plant till I retire.
Along with Rajesh, over 6,000 Himalaya employees worldwide are committed to the company's seed-to-shelf policy. Over 100 scientists at Quality Assurance study the stability of products at every stage of manufacturing and throughout their shelf life. The Quality Assurance team ensures batch-to-batch consistency, safety, and product efficacy. Using advanced equipment like ICP-MS, GC-MS-MS, and LC-MS-MS, our scientists are able to identify contaminants in concentrations as low as 1 part per quadrillion, the equivalent of isolating 1 drop of water from 20,000 Olympic-sized swimming pools. At Himalaya, all our products begin life as a plant. Products typically take between 8 to 10 years to develop, beginning with the identification of herbs and literature survey and all the way to phase 4 clinical studies. The process can take even longer, like in the case of Live52HP, the world's first herbal drug for hepatitis B, which took us 14 years to develop. Our research findings and the results of clinical trials are published in the world's leading medical and peer-reviewed journals. To date, we have published more than 1,000 clinical studies. Persistence.
Okay. Dr. Anasinghe, can you just welcome Dr. Sastry, our keynote speaker?
Yeah, it's really honor. It's really honor to welcome Professor Sastry, but I think Dr. Chandima, my colleague, has a better background which is similar to Professor Sastry, and also his basic degree and also postgraduate all from India. So I gave the chance to Dr. Chandima to welcome Professor Sastry.
Okay.
Thank you.
Thank you, Dr. Anasinghe. Dr. Sastry, welcome you to the 6th International Symposium on Medicinal Plants, Herbal Products and Hydroponics. It is my privilege to give you a brief introduction about your background. Dr. Sastry actually he's obtained BAMS degree at Government Ayurveda College, Vijayawada, and later Master of Doctor of Medicine in the area of Draviguna Vijnana, NTR University of Health Science, followed by PhD at Tilak Ayurveda Mahavidyalaya, Pune. I think we must have met earlier because I also had a chance to visit Tilak Ayurveda Mahavidyalaya during 2005 and 7, where Dr. Nesri was there at the Department of Draviguna. Dr. Sastry has more than 15 books in various disciplines in Ayurveda, more than 50 research papers published, and he owned 8 patents. And also he has obtained Charaka International Award for Excellence in Ayurveda Teaching. At present, he is CEO of JATAS Herbal India, and also he has various R&D exposure in various healthcare sectors, including Dabur, and he is an eminent scholar in the area of Ayurveda in India and elsewhere also. So at this moment, I warmly welcome you, sir, to this international symposium to deliver your keynote speech. Sir, I think, sir, you are muted. Sir, you are muted.
Yeah. Thank you very much, Doc. I'm glad I'm part of today's deliberations, and thanks to the team which has given me this opportunity. We are rather discussing the title of this particular conference, and the theme has attracted me. You are looking at the hydroponics part, and you are looking at the newer techniques and uses of biotechnology. Right now, I'm in Madurai, where I am in a field of about 164 acres, where there are more than 1,000 species of medicinal plants. And this campus trains many of the local healers. And my present endeavor is to look at supply of quality source certified medicinal plant raw material. That's the mandate of my organization as on date. When I look at my exposure, it happened by chance. And when I was in industry, one day I was asked to look at the bioresources team of my previous organization. That's where my focus on different biotechnological interventions and the agro techniques has gone up. And I started focusing more and more into different innovations. And this teaches us that the species which have a lot of genetic diversity are surviving in majority of the conditions, climatic conditions. And those species which have a lesser diversity, they are becoming endemic to specific areas. So this is a small learning. Maybe that's the reason why Ayurveda mentioned probably they understood a particular herb survives in a particular place. Therefore, the people living in that area should better use these materials. Probably that's one concept, but they never restricted the trade of medicinal plants, and the diversity was allowed. So today, when you look at the future, you're all aware, today, India is proud, and Honorable Prime Minister could bring the WHO Global Center for Traditional Medicine to India on 19th of April this year. The center has been established at Jamnagar, which is a big achievement for the entire region, SAARC region. And it's the beginning of the new era. And COVID might have really helped us in this particular aspect, where traditional medicine contributed a lot in this management. And today we are fortunate. The second thing, no science will be flourishing if you don't have a commercial success behind it. Let it be any branch of the science. And today, at a CAGR of about more than 16% to 17% globally for the herbal medicine, has given or opened new avenues for alternative systems or the traditional systems of medicine. So therefore, we have to look at judicious uses of biotechnological interventions in traditional medicine, nutraceuticals, as well as the phytopharmaceutical drugs. Apart from the botanical drugs, there are plenty of plants which have contributed to major medicines, and not to forget morphine or your penicillin or your digoxin. They all have come. Even your cinchona bark resulted in malaria medicine. Artemisinin came from again plant. So we all are aware these plants were the source of some of the major drugs in the pharmaceutical industry. So the emerging market in the pharmaceutical industry is the phytopharmaceuticals, for which again you depend on natural products or the medicinal plants. And apart from that, the traditional systems of medicine, which have contributed to nutraceuticals in many countries, and especially in India, a special system including your Sri Lanka, Nepal, even Myanmar, all these countries, we have Ayurveda as the traditional system of medicine, very well established and practiced for several centuries. So we will start with the collection of the identified plant material, and then we may do an extraction part, or we may use in the whole drug format in the traditional medicine. That is, in the ancient days, we used to call it as decoctions, or the hot infusions, cold infusions, those kind of formulations. And sometimes we were making emulsions like oil-based, ghee-based, and sometimes it is a paste made up of several herbs mixed together and cooked with the base of syrup, that is avaleha, or the linctus, or elixirs. So more or less, your solid and liquid formats, what we discuss mostly in the pharmaceutical industry, were there. And the standards which were laid in the text may be a little difficult today in the modern times, because we have changed the lifestyle. But we have to do the technology transfer properly by understanding the method. Vaguely, if you just pick up a herb and prepare a decoction, that is kashaya in Ayurveda, a decoction, in your own way using an extractor, using the same quantities of material, it will result into a different format. For example, if you pick up a 1:8 ratio of drug herb into water and reduce it 1/4 or 1/8 as per the text in the given condition in the traditional way in a vessel, or use an extract to do the same thing, the total TSS of the resulting material will have a huge difference. If you do it in extract, it will be more than 60, 70 TSS. If you do it in a traditional way, it is 30 to 35 TSS. That means the technology transfer should be appropriate. It should not be involved with whims and fancies. It should be scientific. So those aspects are key and niche areas when you do the pharmaceutical processing. The processing engineering, which is a major aspect in the pharmaceutical industry, should also come into the traditional medicine. And then their standards and their what you call protocols are also very important. So the challenges in the development of phytopharmaceutical drugs, what we have today, are basically when you don't use the biotechnology as a tool per se, you will definitely plan for a particular activity and end up in not getting a activity because initially you thought the whole plant was giving a particular effect, and you have done the fractionation. The fractions may not have. Then you might have gone to the isolation of individual so-called bioactive molecules. By that time, you have ignored the importance of combined effect of primary and secondary metabolites together, working together in a given herb. So that engineering part, process engineering part, is very important. So your extracts or fractions, you should be very clear whether you are mimicking the text or you are making a new formulation and attributing it to the failure to the text is more important. So Egypta herba is one example where vedolactin is taken as a marker and isolation has been done. Various heteroproductive activities or hair growth promotion activity, a lot of things have been attributed. But at the end of the day, when the whole herb is given, what is the benefit we are deriving, and when the isolated compound is given, what benefit is derived, both are to be known. Same is the case with curcumin. Today, with biotechnology and genetic modifications, you may bring in your curcumin to a higher level. I have seen in Northeast, 6 to 7% of curcumin is seen in some of the curcuma longa samples. And now we are getting focused on black curcuma also. So, curcuma acacia. So, when we are looking at these things, we should be really sure what is our requirement. One side, curcumin is not water soluble. So if you are using probably any decoction in the traditional medicine using turmeric, it is already devoid of curcumin, and it must be having other curcuminoids, must be actively participating a therapeutic benefit than the curcumin. So we should be clear what is the actual format given, what is the criteria, and those things are to be taken care. Second thing, before we enter into this particular stage of looking at the markets and all, what is the basic thing that is required when we talk about the intervention of biotechnology in the medicinal plants? The most important thing is the germplasm of the source or the quality planting material which is getting into the field, which is the most important thing today. And NBPGR Delhi and then KFRA Trishur in South India, they have respectively about 120 and 40 species respectively with them, and especially medicinal plants, and their germplasm is preserved and the studies are available. People who are venturing into cultivation or collection, they should be aware what is the actual the plant through which they are going to derive a particular moid chemical entity, which is going to practically. And for that, you need a lot of standardization, and which can come from your DNA fingerprinting along with simultaneous chemical fingerprinting. So genotyping and kenotyping can go hand in hand, and at least we have more than 100 plants experience on this, and amazing. The genetic variations and geoclimatic changes bringing in a lot of variations and corrective procedures. They're all possible today by technological interventions. We always talk about only the RM standardization. RM standardization is the last thing. When you have not taken care of the cause, there is a cause and effect relation. When at the seed level you have not taken the measures which you can take, then you have to suffer when the RM is produced. So many farmers, even when I'm standing here, I hear a lot of problems from the farmers. When they cultivate Withania somnifera, rarely they find sufficient quantities of ethanoids. Okay, people talk about 0.75%, but finding 0.25% of ethanoids itself becomes a great difficult thing. So how do we manage? Because we do not have a hold on the standard operating procedure, there's good agriculture practices, nor we have a hold on the germplasm. So when you don't have that hold, probably you won't get the desired chemicals, or you don't get the desired the therapeutic benefits. So for that, the initial DNA fingerprinting probably is one solution. So the screening should happen for the genetic profiling, and we experience, like for example, when we were working for the US pharmacopoeia, we found Picoraza corova, one particular variety coming from Munshari from Uttarakhand, showing more than 42% genetic variation. And ultimately, this is identified as Picoraza tunganade, as reported by Colonel Chopra in 1956, if I'm right, which was not reported. And later is published in Colombo also. And it has the highest concentration of picrosides, both 1 and 2 fractions. And that is an eye-opener. Till such time, the entire universe is thinking that the Picoraza corova is the only species that is what is available with us. And this continues with many, and many times, if you look at Operculum terpetum, especially in Indian context, Operculum terpetum, by genetic nature and fingerprinting, you find it almost same. But the variety which grows in a particular belt of Gujarat is only acting as laxative. In Ayurveda text, it's mentioned as laxative. So this is because of a specific gene pool of this plant coming from a specific geoclimatic zone. Therefore, always I preferred to classify the medicinal plants and related activities on the basis of the geoclimatic regions than the agroclimatic zones. There are established 15 agroclimatic zones. Basically, I want to tell you about the difference between these two. Agroclimatic zones, usually the agronomy conventional crops like your paddy, your wheat, your sugarcane, your pulses, you may grow in Himalayas, at least the lower range of Himalayas, Kashmir to Kanyakumari, or east to west coast. But the problem is, you cannot cultivate Picotrella curova in south. You cannot cultivate Inla racemosa in south. That means the altitude variations in the medicinal plants should have a different approach and different classification. So therefore, we should divide the entire, at least the region here in India, into 8 geoclimatic zones. And basis that you should explore. That's some exercise what we have done and we understood. And again, developing the clones or developing the micropropagation methods, we should be very careful. I have seen people working on tissue culture of aloe vera, which I feel absurd. It is my personal impression. I may be wrong, but I felt it. When we carefully monitored about the 250 species, hardly 25 would require a serious intervention with micropropagation. Otherwise, everything will get into micropropagation. So you have to prepare the protocols, and developing the protocol is not a small thing. It will take 2 years, sometimes 5 years also. And sometimes the callus-based approach, this is another approach we should really look at. And this I understood while working with Eclipta alba, and especially with Comifera whitey, is so-called gugulu, which is an extremely endangered species. If you have a bioreactor, you can produce callus, and you can have almost all the benefits of the oleoresin coming from Comifera, provided you know which generation will give you sufficient concentration of the gugulsterones. And those are the kind of stuffs which you can learn from your biotechnological interventions, and that will definitely help you. And sometimes you find high pH soils, which are still suitable for medicinal plant cultivation. So sodium tolerant species, when it comes to the medicinal plants, is another aspect, which is purely biotechnology driven. And if you can identify the species like Bacopa, Ecorus, Desmodium, Uraria picta. Uraria picta is one important thing, Plumbago zelanica. They're all sodium tolerant. So even in high pH soils, you can cultivate them. Thereby you will be helping the farmers and the communities. And this can extend to anything like your DNA fingerprinting along with chemical fingerprinting will give you leads in which gene this plant behaving in which way and which chemical is present, which chemical is absent. So basis that you can the area of cultivation and what is the best the geoclimatic zone in which you should cultivate. So that's where exactly your biotechnology is really going to help. Even it will help you in a different way. Like if you take honey these days, going to another end, you can still do the DNA fingerprinting of their pollens. And you can also look at molecular docking today. After molecular docking, a lot of new research avenues have been opened for your COVID. Most of us, you must be aware, like all the spices. A lot of research has begun in various areas. Andrograph is another one through which the COVID has been managed in some countries. So this is possible because of the molecular docking, and it's a low-hanging fruit from where you can again look at the metabolomics, what kind of primary metabolites and secondary metabolites are there and how to use them judiciously. And then comes ultimately your RM, what you call management. Even in RM management, many times we talk about DNA fingerprinting, especially for the raw material. I felt at the field level, it's very difficult at this point of time to do the DNA labeling. Maybe in selected species you may do, but otherwise your morphological identification and macroscopic, microscopic identification, your pharmacognostic studies will definitely help. And DNA fingerprinting at its role should be minimized when it comes to the RM testing. That's my personal opinion. because fit for purpose approach. One of my friends used to say, since we have all the gadgets with us, I can't go for LCMS. LCMS I want to use when I want to find out whether it's in Chyawanprash, whether it is ascorbic acid or whether it is really the L-ascorbic acid. The ascorbic acid and HPLC, what you report usually is hydrolyzable tannin, but not ascorbic acid. This you will come to know once you take it to LCMS and test for L-ascorbic acid, and it will be negative. So the fit for purpose approach, we cannot use each and every tool for testing the standards. And coming to the latest techniques of hydroponics, like in Hyderabad, somebody I visited is doing Bethania somnifera and hydroponics, and very healthy material and high content of ethanoids and wonderful. But again, each species cannot go into hydroponics. And I've seen in Palampur, they've tried hydroponics for even Crocus sativa, your saffron. It may not be successful, but it is a good attempt. But instead, always I feel this will be useful for plants like tagar, your Sugandhibala, what we call it as Valshi, which is not a successful crop when you look at the field level. People may claim, but it's not a successful crop when it comes to commercial yield. Maybe you may have to look at hydroponics or aquaponics. Or aeroponics is also a technique which is again being used at various places. These new techniques, with a comparative study, standalone may not help. You have to take the wild samples, the normally cultivated samples, and hydroponics or aquaponics or hydroponics cultivated samples, and compare. And another species which can really attract hydroponics for me is Hemidismus. Slowly, industry replaced Hemidismus with Decolapis. But Decolapis gives more vanillin and gives good fragrance than giving good quality medicinal properties. Whereas Hemidismus is having more medicinal properties and more chemical moieties. So, because of the operational aspects where the labor-intensive cultivation happens for Hemidismus, or even wild collection also requires a lot of labor, we have resorted to the alternate species, which is incorrect. So in those areas, if you implement the hydroponics, it will really help. Okay. And all these things should be supported by their genetic studies as well as the chemotyping. ICMR has done a lot of work on the standards. CCRS did a lot of work, and APA volumes have given standards. So we have to collate, and already harmonization is happening with the Pharmaceutical Laboratory of Indian Medicine. So efforts are there. Combining these efforts along with the available what you call biotechnological interventions, judiciously using the plant hormones as and when is required, not at every place. In some places, yes, definitely they will help. Of course, as a separate entity, we can discuss sometime later. What are the plants where you can use the gibberellins and likewise other plant hormones. So we can definitely use these new techniques judiciously for the medicinal plants cultivation collection also. And while collecting also today, NPOP is possible, that organic certification, provided you follow certain norms. And I wish this conference will definitely bring in more thoughts and come out with a lot of new ideas from the young generation. And with this, I would like to stop myself here. And these points which I wanted to share with all of you. I once again thank the organizers for allowing me to share my points with you. Thank you, one and all.
Thank you very much, Dr. Sastry, for your informative talk. And I would like to have a few more questions from your talk. I have drawn that Hemidesmus indicus And Decalapis hamiltoni, you have observed some hydroponic technique in cultivation sites. So I would like to know, these are substitute in according to Ayurveda. Now what we see in Sri Lanka, very thick tuberous climber, that semidesmus indicus, so it is something else, Decalapis hamiltoni or something else.
Decalapis. What you are saying is Decalapis.
This is called Krishna Shariba in Ayurveda, I suppose.
No, actually, there are 3-4 varieties. One is semidesmus, the other one is echinocarpus, which you come across in the north, and Decalapis is more in South India, Nanari, and it is abundantly cultivated, easy to propagate also, gives good yield and a good price in the market. So many times the commerce will dominate the science, and this is one best example. Right. So through hydroponics, if you grow, it is not labor intensive, then you can prune the roots as in when you want, and then you can continue. So that's my thought process. Judicious uses of agrobiotechnology. So I want to precisely say not biotechnology, agrobiotechnology is the right phrase to put. And this is the interface I used to always say, this is the kind of interface we should use from the science to make this traditional medicine rich and commercially viable. Okay, thank you, sir. And thank you very much for your vast knowledge you have obtained in the area of science, starting from BAMS, MD, PhD, and you have moved to your exposure to industry, Ayurveda industry, and even sometimes as a regulatory person in the industry, and you have been very good resource person in the area of Ayurveda, modern Ayurveda, pharmacognosy, Draviguna Vijnana, and science. Thank you very much for your informative presentation, sir. Thank you. Thank you. Thank you, one and all. And now